thanks to one of my favorite institute, because this is my hometown institute where I born, where I studied in Virudhunagar district. So I'm very proud and happy to give talk to this institute, Kalasilingam College of Pharmacy. Thanks to Professor Venkatesan, very great person, very good team organizer. Thanks, sir. Thanks for introducing me. Okay.
My pleasure. My pleasure.
Okay. I really appreciate your giving opportunity to talk to young students, because this talk I mostly prefer for students for their research careers. Also, I surely support your institute for research collaboration and MOU and developing research project for students and knowledge sharing. So I will support through my institute from South Africa. Yes. Okay.
Thank you, sir. Thank you, sir. Very kind of you, sir.
Yeah. Now, okay. My talk will be, I like to interact with the student through basic science, because I don't want to talk like I did big things, I'm doing these things, because this is my people. So I like to talk friendly. So if you have any doubt through my topic, even out of my topic, if you have any doubt about research, like project, how to start research, how to do project, so please open to talk to me. I will share my experience through this talk, because I am also same like you people. I came from very ground to earth. I learned something, so I like to share my experience through this convocation. Okay. Before I go to directly my topic, I like to share about where I am here, where I am in South Africa. I am currently working in University of the Free State in center of South Africa, the city called Bloemfontein. So Bloemfontein is a wonderful city, like bloom, like everywhere you can see rose flower in the road, street. So that's called Bloemfontein in South Africa, one of the favorite city in a small city, also a very clean city in South Africa. So that city, it is the University of the Free State in the center of the city. So I'm working with the Department of Chemical Pathology in the School of Pathology, College of Health Science, as a medical scientist. Actually, I'm working with also, I didn't mention that one. Recently, I appointed as a medical scientist with the National Health Laboratory Service, which is a government lab, as like we calling as a NHLS, like a National Institute of Service, like helping to medical college. It's attaching with the medical college. Okay. So I will go to my topic. Today I'm going to discuss functional bionanoparticle for cancer therapy. Okay. Here, what is the meaning of functional bionanoparticle for cancer therapy? Functionals, we doing something with that surface or nano. I hope everybody know about nanoparticle. I will explain below the following slide. Nanoparticle, what is the bionanoparticle, and what is the functional. Okay, I will explain one by one. So please follow with me. If you're not understanding, you can chat with me. Okay, I will explain clearly. Okay, I'm going next slide now. Okay, this slide I'll tell about briefly about my research career, where I did master and where I did my PhD, and then where I learned research methodology. Okay, I did undergraduate in SPE college in Arpukotai, BSc chemistry. Then I moved to Bharatiya University in Coimbatore, Department of Chemistry. I was working with the Professor P.S. Mohan. He's one of the famous researcher in southern part. He's very expert in nitrogen heterocyclic chemistry field. He's very humble, very honest professor. So this is the platform I learned research methodology to develop my career. Now he is retired now, so but he is a ground to earth professor. Then I got a PhD in South Africa. Then 2009 I moved to South Africa. I did my PhD in Durban University of Technology under Professor Arm Gangan. Okay, he also is born in South Africa, but he settled in his family came from India 100 years before. He settled in South Africa, so he also Tamil family, but he little bit will talk Tamil, but he is a very good professor. So I did my PhD with him. He is my mentor. Also, one of my co-supervisor, Professor Anil Chattergon. He is very expert in medical biochemistry. Recently, South Africa government he gave interview to media. Whole South Africa is his lab helping COVID-19 for testing COVID sample. So he is the lab got PCR instrument. So he did lot of testing for COVID in patient sample. So he gave interview recently. Okay. So I did postdoc with Professor Anil. 4 years I was with him. So I learned a lot of biomolecular work with this lab. So from that lab, I moved to University of Free State now. Okay. So this is called the Durban University of Technology and the University of the Free State, University of KwaZulu-Natal is in Durban now. Okay. Okay. Now I will tell about what I did so far. My field is chemistry. Then I moved to organic chemistry, synthetic chemistry. Then during my PhD, I converted to organic synthetic chemistry into nanochemistry. Okay. So basically, during my master, I was working with a plant-based compound, isolating compound from plant. Then I tried to synthesis in the lab. Then I tried to modify with the nano formulation. Then I started testing with different cell lines like cancer cell lines and diabetics models and HIV models. So these are this slide you can see. I edited one book. You can see during my PhD, my university newsletter, I obtained the newsletter. I got best doctoral student research shows huge potential for cancer treatment. It came to my newsletter. Yeah, these are the compound. These are just a small. Intro about me. My most of the research work based on microRNA and DNA. So I designing molecules to control microRNA and DNA. This was my project before. Okay. Okay. This is nitrogen heterocyclic chemistry, which I learned Professor Mohan lab, try to synthesis and designing the molecule to control the DNA, because the DNA is a very important role in a nucleus. So we try to design the molecule to control the DNA, like inhibiting, activating. So I designed some molecule, let's call culine based molecule. One of the molecule ellipticine, cryptosine. So these are the molecule we design and we synthesis. It was my master project. Okay. Then I moved to South Africa. I started organic chemistry. When I came here, I'm the first student for my professor. So that lab was a phytochemistry lab. Then me and my professor developed that lab to organic chemistry to nanoscience to medical biochemistry. Okay, so it was very good experience, because when I came to lab, just a few stirrer and some column was there. Then after 2 years, I developed that lab. We wrote grant for South Africa and South Korea funding. Then we got 50 lakhs grant. Then we bought a lot of equipments. Then things we developed lab, very international level quality lab. Then PhD student, we brought it PhD student and master student. They all are settled very nicely now. Okay. Now, before I go to my topic, I will you can see the video, bioconjugated nanoparticle. You can see here, the center of gold color is a nanoparticle, so functioning with a different molecule. So the different molecule, drug molecule, are like antibodies. They're delivering to human clinical trial, like a clinical study. You can see there is a delivery to blood vessels, is targeting to tumors. So the tumors is like a reducing through this formulation, like the conjugating nanoparticle formulation. So why we here we using the nanoparticle and the functional? Why we doing here? We try to reduce the cytotoxic unwanted side effect through the treatment. So this is so we need to target like receptor target. So we need to target exact area. So we need to functional based on that tumor biomolecules. We need to design the molecules. So it's not going to give side effect to normal cells. Okay, this is the concept they are applying for nano drug formulation, nano drug delivery. Okay, so this animation video, I think maybe you enjoy this video easily. Instead of studying the slide, you can understand through this video. Okay, now, okay, mostly I work with gold nanoparticle. During this work was my PhD work. So I choose gold nanoparticle and the sulfur interface. Okay, because we cannot bind all the molecule in nanoparticle surface. We need to choose proper ligand and proper surface modifier. Based on that molecule only, we can functional our nanoparticle. So nanoparticle is here carrier. Functional molecule is our drug molecule or our antibody or whatever DNA, RNA. So these are our delivering molecule. So nanoparticle is here carrier delivering molecule. So you can see the difference. So the delivery molecule, we need to function with the sulfur, because the sulfur and the gold easily can interact. These are some chemical bond between sulfur and gold. So easily can interact. So this is called sulfur deviate covalent bond. Because we can easily attach with this gold surface, we can easily remove. I mean, it can release easily. It's not make strong binding with the metal surface. Okay, so we choosing the sulfur. Okay, so these are the sulfur and the gold concept. So I use most of the functionalization method sulfur based molecule. So for example, I'm taking common molecules A. I modified with that A sulfur functional group like thiols, disulfide, thioether. Then I will bind with the gold surface. Okay. You can see one of the work from my PhD work. Synthesis of novel carbosol thiooctanic acid capped with gold nanoparticle. You can see that molecule that called CTN here. You can see this molecule is a very good DNA intercalator that's called carbosol. So I react with the lipoic acid which is containing disulfide. This molecule I synthesis in my lab. Then I created a gold nanoparticle. So I function with the gold surface. So you can theoretically image, you can see here the gold nanoparticle top of the surface. You can see the drug molecule attached here. So these are the TEM image of the functional nanoparticle. So this work we done and we submitted to scientific report. Article is under review now. Okay, you see here possible mechanism for formation of that CTN capped nanoparticle, which I discussed before, sulfur and gold surface is a covalent bond interaction, like slightly, it's not like a strong interaction, it's a weak interaction. So it will bind, also it will release. Okay, depends upon the surface, that molecule surface, atmosphere, like based on the protein enzyme reaction. Okay, so we tested on cancer cell line, that's called the A549 cancer cell line. So it's a mitochondrial accumulated transition MTT assay. So it is damaging the DNA. So it's cell get dead. So we did some biological work on this bioconjugate nanoparticle. Okay. Then I moved to self-assembly organic nanoparticle. So I used that one gold nanoparticle to deliver that carbosol molecule, but this is totally different from which I did previously. Here, no metal nanoparticle, full and full organic nanoparticle. See, I did a lot of work, different kind of work, but I'm explaining on this slide is saying some of the important work only. Okay, first I used gold nanoparticle to deliver my small drug molecule. Then what I did, I don't want to use metal nanoparticle. Then I started we stay on organic nanoparticle. We do some work with the organic nanoparticle. Then I started. They are different organic nanoparticle, polymeric nanoparticle is there, but I cannot bring all the different field to my lab. So I need to design based on my lab, what I have, what we can do. So you can see here, same, that organic. I took carbosol, aminocarbosol, and lipoic acid. This is a self-assembly, means nothing but is assembling nanoparticle itself. Okay, so how it is assembling? Okay, we need to think about some physical properties. Okay, so what I did, I take water soluble compound and a water insoluble compound. So like lipo, like you can see like a lipoic interaction, like a water insoluble and water soluble. Then I react together. Then that molecules containing plus or minus like that. Okay, water soluble and water insoluble. Then what I did, we dissolve with the solvent, which is dissolving this properly. Then I mix with the insoluble water. For example, if this molecule dissolving in ethanol, I use non-polar solvent. Okay, polar non-polar combination. Then I did emulsion method. Is creating nanoparticle itself. Okay, this is like an organic nanoparticle. You can see here the tame image of my nanoparticle. So that carbosol and lipoic acid both are very good biological active. Carbosol is a aminocarbosol. Here aminocarbosol is a very good DNA intercalator. The lipoic acid is a very good antioxidant. So my aim to, we need to target DNA. We need to improve mitochondria activity based on. Okay, so this one kind of good one molecule have good properties, one molecule have bad. I mean, like act as a drug, one molecule act as a nutraceutical. Okay, so we don't want, see, it's the same molecule, we don't want to disturb the normal cell. So that's why I designed this molecule like. You can see the caspase mediate apoptosis in HP2 liver carcinoma cells. So this is small graphical picture of the whole work. We did caspase 9 and 3 and 7 caspase is getting the cell get death. So this molecule is very good for cancer studies. But if you take the same molecule separately as a control, we're not getting that much result. So if we combine together, we creating nano formulation is more effective than the normal single molecules. Okay, so because of the nano, why we saying here nano formulation, it's a very small, so it easily target that cell lines or that target molecules. Okay, so that's why we're going for nano formulation. Okay. Then a small like bi-inspire nanoparticle. which I did my PhD. We same thing, like we isolating compound from plants, uh, like active compound. Mostly I prefer for moringa plant because it traditionally we have very good impact on healthcare. So I choose moringa oleifera. People used to take this from as a nutraceutical, but still people need to understand how to use this plant. Okay, what is the molecule is very important molecules. Sometimes when you cooking time, when you processing time, we losing that molecules. So we need to understand these molecules in the plant to secondary metabolics. We need to understand metabolics, which metabolics. I mean, metabolics mean nothing but is a secondary metabolics. There's compound, biological biomolecule compound, which is very important. Sometimes it's coming like a essential oil, like a moringa oil. Sometimes moringa crude extract. So there are different type of way of methods to isolate the compound. Sometimes moringa sprout. The sprout is different from a whole plant, more effective than the whole plant. So they are different. This plant is very interesting plant. Okay. So we did this work with the nano formulation with the moringa plant. It's a very good result we got, like a moringa protein. Also, we isolate moringa protein from moringa seed. We developed a nano formulation with the moringa protein. So we did 2 years. We did some very good work with this plant. Okay. So this is some of the work I did with some plant. It's called Tulasi. I did some of the work with the metal nanoparticle. It's the same like previously I discussed about biosynthesis nanoparticle. Then we did a diabetics work also. Yeah, these are some result we have. We published this article with this channel. Yeah. Okay, now you can see here how the plant molecules interact with the nanoparticle. Because of the molecules, we need to choose the molecules which is have oxidation reduction properties. Okay, so then only we can formulate with the nanoparticle. We can create nano formulation. Okay, so I mostly prefer plant have flavonoids, something like which is have oxygen molecule because it is very good antioxidant. So I choose that kind of plant. I isolate the plant. Okay. Now, then after that I move to nanoparticle protein interaction field. So I synthesis nanoparticle. I formulate with the drug molecule. Then I try to bind with the protein like a human serum albumin. So we study protein interaction study. Then we publish some article with the process biochemistry. Yeah, some of the very good journal. So this work, protein binding studies, as well as theoretically we study about how the nanoparticle binding with the protein surface. Okay, is it damaging or I mean is it incubating or activating? So these are study we did through this work. Okay. Okay. So far I discussed about what I did in my PhD. So I started through synthetic background. I synthesized a lot of molecules. I saw two type of work only: golden nanoparticle and self-assembly work. So I did same kind of work, different kind of culanine, carbosol, coumarin, different type of molecules. So I didn't show all the things. So synthetic chemistry, then I converted to nanochemistry, then I converted to biological chemistry. Okay, so three field I did during my PhD because my supervisor is basically organic chemistry. One of my co-supervisor is basically molecular biochemistry. So I learn both people, like both field, like chemistry as well as molecular biochemistry. Okay, then after my PhD, then I started a research career with that's called exosome. Okay, the exosome is nothing but, so I was working with the nano formulation, nano medicine, something. So when I joined as a postdoc with the Professor Anil lab, we just every week we have a journal club that's called journal club. We every week Friday morning we have discussing group discussion. So we was discussing about mycotoxin. Actually, that group mostly working with the mycotoxin cell pathway, how the toxin doing pathway mechanism with the normal cell and the cancer cell. Okay, for example, mycotoxin nothing but the phosphoric acid, aflatoxin. We every day we eating food, but we don't know that food is contaminated with the mycotoxin. Okay, for example, we eating bakacholam, cholam we eating. So we don't know. Sometimes they keeping the store for long. They're not putting the expiry date. So you don't know the mycotoxin fungal will grow naturally. So we cannot see visibly. So but our people are very innocent. They will buy food, they will buy vegetable, even they will buy, for example, tomato. They will buy. They not seeing properly is like is a fungal attack or is a normal fruit or vegetable. So our people will eat directly. So that fungal containing mycotoxin. Okay. So my professor mostly their lab full and full setup with the mycotoxin research. Okay, the mycotoxin how cell pathway with the how is creating cancer, how stimulating the normal cell to cancer. So this was that lab project. Okay, I mean the whole lab they set up based on that mycotoxin toxicity research. Okay, so I entered the lab, then I started a nano drug delivery project there. Then we had discussed every Friday. So that time I was did research experience with the biosynthesis nanoparticle, chemically synthesis biosynthesis nanoparticle. Then one time we discussing about, because I'm new for clinical for that lab. So they are discussing. So now you can understand they are synergetic knowledge exchanging here. And why I'm saying about my lab, I'm a chemist, pure chemist. They are biological chemist. So we discussing the group discussion. So we sharing that experience. That time we getting new field. Okay, I don't know about the biology. They don't know about the chemistry. So we discussing about project. Then they was working with the mycotoxin that called phosphoric acid, aflatoxin, but they don't know what is the mechanism behind that protein and the mycotoxin interaction. But as a organic chemist, as a chemist, they know, okay, this molecule, okay, binding with this protein site, so disturbing this amino acid. That's why the protein got damaged. That's why the K space is increasing, K space is decreasing. So we explain like that. So we have very good teamwork. So they also understanding, I am also understanding clearly. So we have, so this way always the biochemistry people, very good research people. So if they interact together, they will get very good result output with the proper knowledge. Okay, some people do research, they will get result like a machine like that. They put A and B, they will get the C. Okay, they write article. Okay, they publish. Not like that. We need to understand what happening with the cell and the molecules. Okay, so one of the journal club they're discussing about something called exosome. Okay, because they are clinical people. Then I ask why, what is the exosome something? They're saying exosome is just like a some biomaterial from like look like a blood, red cells, white cells, same thing like they're saying. Then I read about exosome a lot. Then finally I'm seeing exosome is nothing but exosome also biosynthesis nanoparticle. Okay, it's a naturally human producing this nanoparticle through our biofluid like a blood, urine, milk, saliva, everything containing exosome. Okay, it's very interesting. So I study about the exosome. The exosomes is like look like a spherical. So the size is 30 nanometer to 100 nanometer. So it's a proper biosynthesis nanoparticle. Then I realized what is the real biosynthesis nanoparticle. So which I did in chemical synthesis, not a biosynthesis nanoparticle. That's a chemically synthesis nanoparticle. This is the real biosynthesis nanoparticle which I learned from that lab. Then I tried to establish that project with my chemistry background. So we did some work in the lab. It's very good work we did. We isolating exosome, we characterizing the exosome. Okay. So now you can see the exosome. Okay, the exosome is nothing but extracellular vehicle by gene. Okay, okay. Now you can see here exosome. So look like a father and a son like that. Father, how father like father, mother, like a son and a daughter. Okay, so exosome carrying genetic material from the parent cell. Okay, the cell giving message to the exosome. Okay, take this message to another cell, give that message. Okay, so the exosome is containing the message inside through microRNA, DNA, the protein, everything. It's made up with the protein. It's containing microRNA and DNA, same like mini cells. Okay, so if you characterize the exosome, we can find out a lot of biomarkers. Biomarkers nothing but disease biomarkers. Or for example, one person have cancer, how spreading to normal cell. Okay, who spreading? Okay, who's that thief? If you see that one clearly, the exosome taking message to normal cell. So this is, I will explain all the thing in the following slide. Okay, you can see here the exosome how transport to one cell to another cell. You can see here this is one cell. When releasing the exosome, the orange color spot is the exosome. You can see the small tube like that. It's called nanotubular. So see, we working with the exosome only that the spherical. Some of the researchers are working with the tube. Like you can see the cell line, you can see some microscope image, you can see some fiber like that. That's called nanotubular. So the exosome traveling through one cell to another cell through this nanotubular. Okay, so this is very amazing. Okay, so it's not like see here you can see like a courier service like that. So the courier parcel is the exosome, the tubular is the road. So one place to another place is transporting. Okay. Okay, now we can see this video. You can see this video is a real video through. Okay, you can see here the cell how communicating. The cell communicating through the nanotubular small tube. So you can see the communicating, the talking each other. So the exosome, the bright particle traveling to one cell to another cell, then means something message passing through one cell to another cell. Okay, something good or bad, something message taking to one place to another cell. So you can see here, figure here, you can see the tube particles. Then the cell getting dividing, so something happening. Okay, maybe cell growing, maybe cell dying. So related to all the disease, this exosome related to all. Okay, now this exosome is a cell-to-cell communicator. Okay, not only cancer, most of the disease, most of the body metabolic syndrome, body action, everything based on the exosome. Okay. Um, See now, then we started with this project with I tried as a chemist. I tried to develop project with the exosome protein inhibitor, because the exosome protein have some functional like the exosome surface protein interact with the other cell line. So I designed project based on exosome protein binding molecule. I designed some molecule to bind that exosome to stop that function. Okay, for example, one cancer exosome taking that message to message from the cancer cell, then taking to normal cells, then the protein function doing some the transport uh, function. Okay, so my idea was to stop that protein function, then you can control that exosome to transfer message to normal cells. So this was my project. So we recently published this article with the AC Sumeka. Okay. Okay. Now, what are the application for exosome? Okay. See why I'm telling about the exosome. You people are a very young researcher. Instead of uh, thinking different project, it's my advice to, to start up some project with the exosome, because it have in the future regenerative medicine is a very important. Okay, we need to find out the proper disease biomarkers before developing some drug molecules for disease something, because still we not understanding about disease, why happening, how's happening, what's the reason. Still we not understanding about proper cell mechanism and the cell pathway, protein pathway or enzyme pathway. So still we not understanding proper mechanism between the cells. So that's why still we struggling to find out medicine for cancer and HIV and Alzheimer, because still we not understanding about disease biomarkers. Okay, some people find some biomarkers, all the research are focusing on that biomarkers. Okay, they found something. For example, COVID-19, some people find out the spike protein, all the people talking about spike protein. Then some people find out protease protein, then all the people talking about protease protein. Still we don't know COVID virus containing different other protein or something different. We need to find out the proper biomarkers or functional protein functional. Then only we can think about drug designing or vaccine designing. Okay, so I'm believing every disease the exosome is playing very important role. Okay, so if you isolate the exosome, if we characterize the exosome properly, in future we can design the drug molecule or anything based on the exosome biomarkers. Okay, so these are the application. Exosome we can use as a diagnostic field. Exosome we can use as a drug delivery field. Exosome we can use as a target. Okay, target means like a protein. We can develop antibody, monoclonal antibody, something for vaccine. Okay, biomarkers, diagnostic field, everything biomarker. For example, if you're going to do blood test to check glucose, the glucose, the enzyme is one of the biomarkers. So that is a well-known method. But in the future, in the future unknown disease like in the COVID-19 virus, something we try to find out new biomarkers. Okay, so the biomarker is very important one for future. Okay, drug delivery. Okay, so because this exosome is a nucleic nanoparticle, so we can use this exosome as a drug delivering agent. Okay, instead of using different type of molecules or different type of nanoparticle, we can use this exosome as a drug delivery agent, because that protein surface know which the protein, which cell surface we can interact, because it naturally is made up with in human body, so it know. So we can use the exosome as a drug delivery agent. Okay, see now you can see that this field exosome, this slide explaining about the exosome diagnostic. Okay, see now why we want to do diagnostic for exosome. Okay, see now currently we are working on pathology lab, so understanding about diagnostic, how they are doing diagnostic. There are different protein, different enzyme function, different secondary metabolics. Based on that one, they are designing the biomarker kit, the diagnostic kit for finding disease. Okay, so saliva, blood, urine, all the thing, body fluid containing. For example, current method is they're taking blood sample, and the doctor will say takes test, blood test, urine test, everything. Then they will come to our lab. They test. We doing some assay test like a vitamin analysis or whatever the patient have disease. For example, the patient have acute kidney failure. Okay, so the doctor will advise, okay, do kidney stone analysis or do this blood test. So they will come, the sample come to our lab. We do kidney stone analysis. We can find out what is the protein or what is the inorganic material in the kidney stone. So we can say, okay, these are the disease because of this protein express, because of this enzyme, because of this chemical, secondary mechanism, this patient have acute kidney failure. Okay, so these are some example I'm telling you about. Same thing in future. So this kit, for example, if you identify acute kidney failure, we need kidney stone. Okay, so in the future we need to find out simple way to find out any type of disease. It, instead of taking blood from patient, it's like a painful to take blood than a testing. For example, okay, big people no problem. What about the baby? If I like a 5 month baby or a 10 month baby, even a 2 days baby, you cannot take blood sample from baby. Okay, but you can take saliva. Okay, so we need to find out proper, I mean, a easy diagnosis method for future generation. So the exosome currently is very important role. For example, exosome, we take saliva, we isolate the exosome. If you do characterization of the exosome, we can find out, okay, this baby have this syndrome. Okay, like instead of taking blood or something from the baby. Okay, so exosome is future diagnostic material. So okay, then now see here, exosome target delivery. Okay, so I told before we can use this exosome as a drug delivering agent. Okay, because the exosome naturally containing some protein. That protein is made up with the human body. It's not like artificial. It's not like out of our body, like a synthetic body. So it is already in our human body. Because, for example, I'm simply saying, I will show one slide next how the future treatment will come. Okay, for every single personalized medicine, how it going to come in future. Like a personalized medicine means, for example, one person have like a small age to up to 50 years, he have this diabetes, Alzheimer something. So they have variety of I mean data of his whole life disease profile. So we can think about, okay, this person have this disease, that's why he's getting. So we can think that's called personalized medicine. Okay, so same thing. So we can use as the exosome as a drug delivering agent for future drug manufacturing. Okay, you can see here. This slide explain about exosome biomarker selection for immunotherapy or whatever or any therapy. Okay, I mean HIV or cancer or anything. You can see the patient is the center of the patient. Okay, the patient, we know the patient profile. The patient have disease. So we taking exosome from the patient, like how can say, the patient have the exosome, so we can understand easily through the exosome what exactly the patient metabolic body metabolic and disease biomarker we can understand, instead of taking standard from other protein or other outside somebody made reference. We can take this directly from the patient. So we isolated the exosome. First we characterizing the exosome, like biomarkers what patient have. So we can use that same exosome as a drug delivering agent. So we can give back to the same patient. So these are the future treatment. Like as you can see here, like quality control, exosome isolation, purification, drug loading. Even you can see therapeutic protein, genetic material identification through the patient exosome. So this is the future treatment will come like that. Okay. Okay. Now, so far I explained about my research field. Then currently what I'm doing pathology lab. Now just this slide current situation, how we can correlate with the COVID-19. Okay. So the exosome with COVID-19, currently people are starting project now. Recently, Israel Institute, they set up in the project with the exosome with the COVID-19, and then US also. Because why still we struggling to find a medicine for COVID-19? We don't know proper biomarkers for COVID-19. Okay. So people are start designing the project based on exosome. If you identify the exosome from COVID patient, or the exosome may be carrying novel biomarkers for this disease, COVID-19 disease. we can design the vaccine, or we can design that drug molecule based on the exosome biomarkers. Okay, so people are starting the project. So I hope we will find some medicine for COVID-19. Okay, because of, if we compare with the exosome and the COVID, it's more or less same, like the size or the diameter, the receptor, ACT receptor is containing RNA, is found in the lung fluid. Okay, for example, if you take COVID patient the lung fluid, if you isolate exosome from the lungs fluid, it carrying proper biomarkers for COVID-19. If you isolate that proper biomarkers, microRNA, all the thing, genetic material, then we can develop a vaccine for COVID-19. Okay, or we can design the molecules for drug molecule for COVID-19. So this is the future, not now. Future will come like that. Okay, you can see here, yeah, exosome and virus, both how they is the virus also same, like it's coming from a fluid. The exosome also coming from the fluid. Okay, because I'm telling the virus, I'm telling about inside the body. Okay. Now you can see here the exosome companies. Okay, young researcher and who want to develop the exosome project. Most of the people, we like everything is a company, or what is the product we're going to make it in future based on all our research is linking with all the thing product level. Okay, so recently exosome based company is booming up now. All the pharma company, the traditional therapy, they changing to exosome therapy. That's called stem cell therapy, regenetic genetic medicine. So they set up in all the company based on the exosome. Most of the big company, they're changing their the core R&D to exosome research. So traditional chemical antibody development in the future going to change. So most of the company, they're going to set up with the exosome medicine and exosome diagnostic. These are okay. These are my reference. You can see some of the reference. Okay, now I'm final stage of my slide. Okay, thank you for listening my talk. Okay, so these are the funding agency from South Africa. That's called a National Research Foundation. This is a main funding resource source from South Africa. In future, anybody want to do PhD or postdoc, you can check this NRF website. There are a lot of foreign opportunities there. You can come to South Africa, do research and higher studies through this funding. Okay, because most of the people, especially our Tamil Nadu people, always thinking about Taiwan and South Korea, or some like US something. So South Africa also a very good country for higher education. It's an English country, English based country. It's not like Africa means something like forest. I can say when compared to India, South Africa well developed. I mean not knowledge wise, like specialty institute, higher institute, all the things like infrastructure. So everything is so. You can come to South Africa, do higher studies, do research. So you can contact me because I'm 10 years in South Africa. I know all the loopholes here. I know all the things from South Africa, which institute, how can enter in this institute. Because here all the thing government institute, not a private institute. So is everything proper channel. If you apply properly, if you find proper supervisor, if you find proper lab, you can enjoy your research in South Africa. Okay. Okay, I put my question myself. I hope I will get some question from you people. But anyway, from this slide, you can think about what is the bioconjugate, what is the difference between inorganic nanoparticle and organic nanoparticle, what is the gold sulfate interest, what type of bond, what is the nanosome, what is the exosome size, and what is the exosome. So these are my questions. You can work it out. Okay, now it's finished now. Okay, thank you. So
Thank
I
you, sir.
done.
Thank you.
Yeah.
Thank you, sir. Thank you. Any questions?
Alice போட்டாப்லையா? Hello, can I ask you a question?
Yes, yes,
Thank you very much, Dr. Anand, for the nice presentation.
yes.
I
Thank
have a
you.
question. You earlier you talked about this sulfur.
Introduce yourself, sir.
Ah,
Sorry.
First, introduce yourself, please.
okay, sure. I'm Dr. Bhaskaran from University of Western Cape, South Africa.
Okay,
Okay,
okay. Oh,
yeah, I'm just asking about these gold sulfur interface,
yes, yes.
sulfur functionalization. There are also amine functionalization. What about
Yes,
amine
the
functionalization?
yeah, amine functionalization also I did actually. Amine functionalizer is like a 10 trimer or something like that. Amine functionalizer we can do, but I prepare for sulfur because of I mostly use for gold because I tried silver nanoparticle also. I tried copper nanoparticle also, but gold is a very less toxic for cell lines. So when I'm reading the article, most of the work, the gold surface modification, most of the from sulfur compound. So I choose sulfur, but we can do amine modification. Even we can do phenolic modification also, like OH group. Yeah, we can do amine modification also.
Okay, thank you, Dr. Anand.
Yeah, thank you.
Students, raise your question. If you have any doubt, please ask.
Yeah,
So good afternoon, sir.
actually, yes, yes, yes,
Sir, you were born in Arpukote. You studied there. Then you moved to
yes,
Coimbatore.
yes,
We are there in South Africa. Can
yes.
you able to explain the journey to our students?
Oh, okay, okay. Nice question. Because actually, I'm not. I hope students are listening my talk. Uh, um, I actually I study in a SPA colleges. Yeah, actually, I'm a very normal student. Sorry to say people are thinking, okay, foreign means we need big knowledge, English knowledge to travel out of country. It's not like that. Everything hard work. You not believing me? During my BSc in Max, I got 4 arrears. So people are that I'm not going to success my life. So within one semester, I finished my 4 area Max because it's, it's not like I'm a brilliant student, like, but in school life I was brilliant student. I tried to study medicine. The Anna University spoiled my career. For 3 marks I lost Madurai Medical College. Then my life, I really disappointed with the entrance that time. Now people are talking about NEET something, but nobody talking about that Anna University. I don't know what is the entrance exam. In school I got a lot good mark in physics in 199, biology 198, in chemistry 195. I supposed to get doctor, but the entrance changing my life. Then I was disappointed with this thing. Then I told my father I'm not going to study higher studies. Then my father put me in the local SPA college. Then 1 year I was disappointed with this thing. So I got arrear in max. Then why try? I'm trying saying all the student can success their life. Then I moved to Bardiyar. I when I bar there, I got a lot of experience with the different type of people. Even though I study in university, I lack with the English knowledge. Okay, so I cannot talk fluently. I cannot. I know what is the reaction mechanism, everything, but I cannot express. Okay, this is right or wrong. So I study with Rani Mary College student, Trichy St. Job, all are talking very good English, but still I believe myself. I have some confidence. I still remember during my master, the class seminar, everybody thought I'm basically from Madurai. I don't know how to talk because our language is mostly from Tamil. They are talking very English medium student, but this class seminar, I'm the number one student to how to present, way of present, how to express our knowledge through what I learned English. Okay. Then I came to South Africa. You're not believing me. I don't know how to talk. Like I know some words, thank you, good morning, yes, no, something. But the South Africa is a full and full English-based country. So I believe myself I can survive this place through our hard work. So this message, please take student, everything hard work, everything interest is not about we need to study English, we need to do this course, that course. Not like that. Everything hard work, interest, always believe yourself. You can do it in anywhere in the world because our people have, as basically Tamil people have hard work and like dedicating. So if you follow these rules, I can say anybody can survive, anybody can achieve their research career and life. I always believe our Dr. Abdul Kalam, dream. Okay, so you need to dream. Still, I got a lot of dream to success my life. So you need to dream. Don't stop. Okay, we did something. Okay, we done with all the thing. Don't think like that. Life is we need to dream. We need to success every day. We need to survive this world. So my advice to young student, don't think you study in a rural, you don't know out of the box. Maybe you don't know, but you can do it. Take my example. I've got 4 areas. Within one day, within one exam, I finish all the thing. I don't know how to talk English, but now I'm sitting with the Bloemfontein full and full white people. Okay, British community. I'm sitting with the white community with the fluent English knowledge. Most of the people they asking advice to me how to do this one, Dr. Anand. How I'm working with the medical doctors actually. See now my life totally changing now. I supposed to become medical doctor during my +2. I cannot make it, but now I'm teaching to medical students. This is my achievement, I believe. So don't disappoint if you're not succeeding. Work hard, show interest. You can do anything. Okay, thank you.
Thank you, sir. Thank you, sir. So, if you want to pursue a career in US, Australia, so you have to complete GRE, TOEFL, like that. So, whether the
No,
same
not
category is applicable to
no,
South
South
Africa?
Africa not South Africa not like that. Even though South Africa English country, no need GRE, TOEFL. It's all about finding supervisor. Okay, which finding lab. That's why I'm telling the NRF all the time the opening call for do PhD in South Africa. That's called towards NRF. Actually, I'm sorry to say most of the Andhra students coming to South Africa through that funding, but we don't know. Our people don't know because most of the people thinking South Africa is not a good place to study. Maybe they don't know. So I am actually after I came South Africa. When I came South Africa, Tamil people very few of them. Now is getting English now because of sharing our experience, telling information to our hometown people. So it's like that. So I can say South Africa very good for research. They giving a lot of source to develop the research, not like a other place to if you want to order some chemical you need to wait for long the processing something. Here within 1 week you will get all the things. Okay, because they have a lot of funding. to develop their research career. Okay, because it's a developing country, so they're supporting for research. Mostly South Africa working with the foreigner, because here's this country is they want to collaborate with the foreigner to develop multidisciplinary synergetic knowledge between different country people. They want to develop the indigenous people. Okay, so they're supporting nicely. So no need a GRE, TOEFL, only how to apply, where can apply. So if they know the detail, they can get position in South Africa. Yeah.
Okay. Thank you, sir. There is a question in the chat box regarding the scholarship amount. Whether scholarship amount is sufficient to run a family in the South Africa.
For postdoc research, they can, yeah, family means for postdoc they can run here, but not like a big family. Maybe husband and wife, one children or two children, they can run. Depends upon the supervisor support also. That's why I'm telling. If you find a nice good professor, this professor also supporting. So that's why before anybody, that's why from this meeting, anybody want to try to apply South Africa, let me know. I know some of the place, which place is good, which place is not good, because sometimes some of the institute is very rural, so you cannot stay there, you cannot live there. Some of the place is very good. For example, Durban is a very good place for live India, because there are a lot of temple is there, a lot of culture programs there. So you can adapt with the Durban culture. Our family can adapt with the Durban culture. So you need to choose which place is to live. So that's why if anybody want to apply to South Africa, please contact me. I will tell the lab, but because I know all the institute, professor, everything. I mean, I can find out even if I don't know the professor, I can find out through my friend circle. So we can give some info about before they coming to South Africa. Okay.
Thank you, sir.
Yeah.
Thank you, Professor Dr. Anand.
Yes, sir.
Thanks for your support and
Yes, sir. Thank you.
and extraordinary explanation.
Mm.
Sir, your presentation went on very well
Thank you, sir.
and elaborated.
Mm.
On behalf of our management, I hereby express my wholehearted thanks to you and your management for giving such a demo presentation.
Mm.
I hope this interaction will motivate the students and encourage them in active learning forthcoming days, sir.
Sure, sir. I will support you. I will keep in touch with your institute. Not only I like to support your institute because we are from southern part. Instead of helping some foreigner, US, UK people, I like to help you people, because of most of the student like me came from Virudhunagar or Stivaliputur or Tirunelveli, Madurai. Same, like I feel like my family like that. So I like to help you people. So whatever help from me, proper way, proper channel, I will help surely, for mainly for student. Okay.
Thank you, sir. Thank you, sir. Thanks for your assurance.
Okay, sir.
And in future also, let's have a good collaboration, sir.
Sure, sir. Okay. Sure.
Thank you.
Hello, sir.
Okay. Once again, I'm thanks to the institute, especially Professor Venkatesan. I really appreciate his hard work, and his team, organizing committee. I really appreciate. Please carry on with this kind of activities, because the institute will, this very good activity, very good method you people are following, sharing knowledge from overseas to our young people. So it will boost up their knowledge. Okay, so they will get interest. Okay, some people think research is a totally waste of time, and we cannot make financially we cannot stable or life. It's not like that. So research is very good field. So people like us sharing the knowledge. See, I'm happy now. I'm living happy life. Yeah. So you people can enjoy your life through research. You can enjoy the research as well as you can enjoy the family also. Okay. So carry on with this kind of activity, sir. I will support. Even I try to introduce my other friends from other country. I will talk to them. Maybe they will also give very good talk with your institute. They will share their knowledge. They are different field. They can share their knowledge. Is supporting your institute. Okay, sir.
Sure, sir.
Hello,
Let me know.
Somebody, yes.
Sir, good afternoon, sir.
Yes.
Sir, this is Professor Rajabandi from AKCP.
Okay, sir. Okay.
Sir, nice presentation, sir. I have one doubt. You are using for making gold nanoparticles. The gold and the sulfur is very easy to join or easy to broken. Apart from this phenomena, any important role for gold molecules, sir?
Yeah, the gold molecules. That's why I'm telling the gold molecules. Why we choosing the gold molecule is non-toxic. So that if it for example we take as a medicine, our kidney need to dialyze this metal. So if it tried with the other metal like silver or some other metal, so maybe if it is toxicity for maybe it will cure that disease, but we don't know it accumulate in the body organism like a kidney or some pancreas it will stay in there. It will give side effect. Okay, but gold not like that. Gold is inert metal. It's no toxicity. So that's why we preferring for the gold, especially if you take gold nanoparticle, it look like how can say people are saying we can eat like a gold biscuit something. It's nothing but it's a nano gold only, the gold biscuit something, because it will absorb our body, it will release through our urinary pathway. Okay, even it's not giving the side effect to our body organism like a kidney or pancreas or even the stomach. So it will release through our human urinary pathway. Also, if we stay in our organ also, I believe, so far the research proving the gold not toxic to human organism. Okay.
Thank you, sir.
Yes. So I like to get question from students. I think. Okay, I will give my mail ID. Yes.
Sir, good afternoon, sir. Sir, this is Arjun Kumar from AKCP, sir.
Okay.
So actually, I have one question, sir.
Yes.
You have mentioned in your slide in exosomes for immunotherapy.
Yes.
In that you have mentioned you have to separate the macrophages from the patient.